Welcome to the 2009 Cell Signaling Technology in-house training tutorial for the study of stem cells. This tutorial contains 3 sections. Section 1 provides an overview of embryonic stem cells and ESC markers. This section also includes information regarding induced pluripotent stem cells, also referred to as iPS cells, and CST's new StemLight Pluripotency kit. Section 2 covers information concerning stem cell differentiation along specific lineage pathways and corresponding lineage-specific markers. And finally, Section 3 covers epigenetic regulators and marks commonly found during embryonic development. What is an embryonic stem cell? Embryonic stem cells, also called ESCs or ES cells, are cells derived from the inner cell mass of a blastocyst. Looking at the figure on the right, a blastocyst is a structure formed early in embryogenesis, after fertilization, but before implantation into the uterine wall. A blastocyst contains approximately 70 to 100 cells and consists of an inner cell mass, also called the ICM, inside a cavity surrounded by a layer of cells called the trophoblast. Cells of the trophoblast later develop into the placenta and regulate implantation of the embryo. Cells of the inner cell mass are called embryonic stem cells and later develop into the new organism. Embryonic stem cells have 2 unique features. First, they are pluripotent. This means these cells can differentiate into all 3 primary germ layers, namely the ectoderm, mesoderm, and endoderm. Additional information regarding these germ layers will be presented in later slides. Secondly, embryonic stem cells have the ability to self-renew, which means they can replicate indefinitely in the undifferentiated state. Why are embryonic stem cells important? Embryonic stem cells are important for regenerative medicine. ESC pluripotency means that these cells can grow into any cell type in the body. Scientists study ESC pluripotency and differentiation with the hopes to create new, healthy cells and/or organs in diseased patients. For example, new neurons could be created for patients with spinal cord injuries, or blood cells for patients with leukemia. Embryonic stem cells are also important for basic science research. ESCs are studied in basic research to increase our understanding of early-stage human development. The study of ESC differentiation along specific lineage pathways may increase our understanding of disease states and progression. Pluripotency at the molecular level. At the molecular level, the predominant signaling pathways involved in human ESC pluripotency and self-renewal are TGF-beta, which signals through SMAD2 and 3, and FGFR, which activates the MAPK and AKT pathways. The Wnt pathway also promotes pluripotency through activation of beta-catenin. Signaling through these pathways results in the expression and activation of 3 key transcription factors: Oct-4, SOX2, and NANOG. These transcription factors activate gene expression of ESC-specific genes, regulate their own expression, and also serve as HESC markers. Other markers used to identify human ESCs are SSEA 3 and 4, TRA160, and TRA181. Pluripotency and self-renewal are inhibited by the BMP4 pathway, which uses SMAD1, 5, and 8 to block transcription factor expression. The pathway shown is for human cells. Pluripotency in mice involves slightly different upstream signaling, primarily the LIF, leukemia inhibitory factor, and STAT3 pathway, but results in expression of the same key transcription factors. Confocal immunofluorescent analysis showing expression and nuclear localization of the transcription factors Oct-4, SOX2, and NANOG in Entera2 cells. Oct-4, SOX2, and NANOG antibodies have been labeled green. Actin filaments have been labeled red with DY554 phalloidin. Entera2 is a human embryonic carcinoma cell line that expresses the same markers as ESCs. Note the presence of green signal in these cells. HeLa cells are a non-pluripotent epithelial cell line used as a negative control and do not express these stem cell markers. Embryonic stem cell markers. Other markers commonly used to identify ESCs are TRA-160, TRA-181, and SSEA4, which are surface markers found on embryonic stem cells and germ cells. They are surface markers only and do not directly add to stemness or promote pluripotency. TRA-160 and TRA-181 are glycoproteins, and SSEA4 is a glycolipid. These antibodies recognize the carbohydrate group, not the proteins themselves per se. The functional role of TRA-160, TRA-181, and SSEA4 in stem cell biology is not clear. Confocal immunofluorescent analysis showing expression of TRA-160, TRA-181, and SSEA4 in NTERA2 cells. Antibodies for TRA-160, TRA-181, and SSEA4 have been labeled green. Actin filaments have been labeled red with dy554 phalloidin. DNA has been labeled blue using the fluorescent dye DRAC5. Induced pluripotency, IPS cells. Induced pluripotent stem cells, IPS cells, or iPSCs, are a type of pluripotent stem cell derived from a non-pluripotent somatic cell by overexpression of a set of proteins. iPS cells have been shown to share many properties with ES cells, including epigenetic marks and the expression of stem cell genes. Two pivotal studies have shown that various combinations of key transcription factors have been used to obtain the induced pluripotent state in human somatic cells. In the first study, retroviral expression of Oct-4, SOX2, c-Myc, and KLF4 in adult mouse fibroblasts caused the reprogramming of these cells into ES-like pluripotent cells. In the second study, a combination of Oct-4, SOX2, NANOG, and Lin28 caused the reprogramming of human somatic cells into iPS cells. Since then, a multitude of studies have repeated these findings in various cell types. Other studies have focused on the necessity of individual transcription factors in somatic cell reprogramming. For instance, the activity of c-Myc and KLF4 can be replaced by the histone deacetylase inhibitor valproic acid. These studies have shown that Oct-4 is the most important component for the creation of iPS cells because it is the only factor that cannot be replaced. The transcription factors responsible for somatic cell reprogramming function by promoting the expression of genes necessary for pluripotency and inhibiting lineage-specific genes. Because many of these genes control their own expression through autoregulatory mechanisms, the overexpressed proteins can bind the promoters of their endogenous counterparts, jump-starting and maintaining their own expression. There are many functional uses of iPS cells. First, iPS cells are very useful for drug screening applications. Researchers can screen compounds on more normal human cells and can better assess on-target and off-target drug effects at an early point in screening. In addition, compounds can be screened for their ability to drive or halt particular differentiation pathways, thus providing information on points of pharmaceutical intervention in particular disease states. iPS cells also provide the ability to generate cell model systems from patients with genetic diseases and allow scientists to follow disease progression as the iPS cells differentiate. A second use for iPS cells is that they eliminate ethical issues surrounding the use of embryonic stem cells. Human embryos would not be needed and as a consequence destroyed in order to obtain an original source of stem cells. And lastly, iPS cells can be used in personalized stem cell therapy. In the future, somatic cells would be taken from a patient and induced to become iPS cells. The iPS cells would be differentiated to create a cell type needed by that specific patient. For example, hepatocytes for a patient with liver failure. With this technology, there would be no need to worry about organ rejection or an immune response because the stem cells originated from that specific patient and have the same immune profile. New StemLight Pluripotency Kit. The new StemLight Pluripotency Kit contains antibodies against a selection of stem cell markers. This kit can be used to analyze the pluripotent status of human embryonic stem cells or iPS cells. Loss of marker expression indicates loss of pluripotency or differentiation of the culture. Retinoic acid treatment, 10 micromolar for 14 days, causes the differentiation of the embryonic carcinoma cell line Entera-2 along the neuronal lineage. Oct-4 and SOX2 antibodies have been labeled green. Actin filaments have been labeled red with dy554 phalloidin. Note loss of pluripotency markers green in the differentiated state. TRA-160 and TRA-181 antibodies have been labeled green. Actin filaments have been labeled red with dy554 phalloidin. Blue pseudocolor is DRAC5. Note loss of pluripotency markers green in the differentiated state. In the plus RA panels, RA treatment is causing the cells to differentiate from small round cells containing a lot of green signal into the larger elongated red cells located in the center of each panel. The red cells no longer contain the green stem cell markers. In the lower panels, neurofilament L is a marker for differentiated neurons, and the neurofilament L antibody is labeled green. No appearance of this marker in the differentiated state. Also note that the green signal is not present in the untreated pluripotent cells. Stem cell differentiation. Upon growth of the embryo, embryonic stem cells differentiate into primordial germ cells and the three primary germ layers, endoderm, mesoderm, and ectoderm. Each primary germ layer further differentiates along lineage-specific pathways to create every cell type in the body. The figure shown here provides a general overview of the primary lineage pathways. Let's take a closer look at the primary lineage pathways and the specific markers used to identify each one. Primordial germ cells are the progenitors of the gametes. These cells migrate to the gonads where they undergo meiosis and mitosis to produce sperm and eggs. Two lineage-specific markers for primordial germ cells are the proteins Mili and Miwi. Mili and Miwi are RNA-interacting proteins that are essential for spermatogenesis. In the figures below, IF and IHC analysis was performed showing Mili and Miwi expression in mouse testis. In the IF panels, Mili and Miwi antibodies have been labeled green. Pancreatin C11 mouse monoclonal antibody, product number 4545, has been labeled red. DNA has been labeled blue with DRAC5. In the IHC panels, note the presence of brown stain with each of the antibodies. Ectodermal lineage. One of the three primary germ layers is the ectoderm. Cells of the ectoderm differentiate into neural stem cells, thereby creating all the cells of the neuronal lineage. Neural stem cells differentiate into cells of the neural crest. The neural crest is a transient component of neuroectoderm present during formation and closure of the neural tube. These very important cells can give rise to neurons and glia of the autonomic nervous system, as well as bone, cartilage, tendons, and smooth muscle. Neural stem cells further differentiate into mature neurons as well as supporting glial cells. This process is called neurogenesis. A number of markers used to identify neural stem cells and mature neurons are shown on the right. IF analysis showing expression of the neural stem cell marker nestin in P1 postnatal day 1 rat brain as compared to mature adult rat brain. Nestin antibody has been labeled green. Neurofilament L antibody, a marker for differentiated neurons, is labeled red. Note the loss of green signal in adult rat brain as compared to P1 rat brain. Note the increase in red signal indicating fully differentiated neurons in adult rat brain as compared to P1 rat brain. The bottom image depicts IF analysis showing expression of the differentiated neuronal marker neurofilament L in mature rat cerebellum. Neurofilament L antibody has been labeled green. GFAP-GA5 mouse monoclonal antibody, product number 3670, has been labeled red. GFAP is an intermediate filament found in glial cells. DNA has been labeled blue with DRAC5. Mesodermal lineage. The second of the primary germ layers is the mesoderm. Cells from the mesoderm differentiate along two diverging pathways. The first branch of mesoderm differentiation consists of mesenchymal stem cells. These cells eventually differentiate into adipocytes, fat, myocytes, muscle, osteocytes, bone, and chondrocytes, cartilage. A number of markers are used to identify cells descending from the mesenchyme. Mesenchymal stem cells can be identified with antibodies directed against c-KIT, endoglin, TAZ, or twist. Adipocyte markers include adiponectin, CEBP-alpha, CEBP-beta, PPAR-gamma, GLUT4, and FABP4. Myocytes can be identified with antibodies directed against desmin, myosin light chain, and troponin. The features below show expression of two common markers for adipocytes and myocytes respectively. First is IFIC analysis showing expression of PPAR-gamma, a marker for adipocytes, in 3T3L1 cells. 3T3L1 cells are a murine pre-adipocyte cell line that can be induced to differentiate into mature adipocytes with specific treatment conditions. PPAR-gamma is a transcription factor that plays a role in the process of adipogenesis. PPAR-gamma antibody has been labeled red to show nuclear localization. Lipid droplets, which are evidence of differentiated cells, are labeled green using BODIPI 493503. Note that PPAR-gamma only appears in cells with lipid droplets. DNA has been labeled blue with DRAC5. In the next figure, IF analysis shows expression of desmin in rat heart, cardiac muscle. Desmin is an intermediate filament that is found as part of the cytoskeleton in muscle cells. Desmin antibody has been labeled green. DNA has been labeled blue with DRAC5. The second branch of the mesodermal lineage consists of the hemangioblast, which is the precursor to both hematopoietic stem cells and the angioblast. Hematopoietic stem cells differentiate into red blood cells, white blood cells, and cells of the immune system. The angioblast further differentiates into vascular smooth muscle and endothelial cells. Markers of the hemangioblast include CD34, CD133, and VEGF receptor 2. Markers used to identify hematopoietic stem cells are AML1, CD34, CD133, CDCP1, c-KIT, GATA1 and 2, PU1, and SCF. The angioblast can be identified using antibodies directed against VE-cadherin and VEGF receptor 2. In the feature below, IHC analysis shows expression of CD34 in human capillary hemangioma. Hemangioma is a benign tumor of the endothelial cells, which are those lining blood vessels. In this figure, these are the cells of the capillary. Note the presence of brown stain surrounding the vessel tubules. Endodermal lineage. The last of the primary germ layers, the endoderm, differentiates into an endodermal progenitor, which further differentiates into hepatocytes, liver cells, and pancreatic cells. Markers used to identify hepatocytes include AFP, albumin, CEBP-beta, FAS, glycogen synthase, HNF4-alpha, and MET. Pancreatic cell markers are C-peptide, glucagon, insulin, NeuroD, and PDX1. The figures seen here show expression of two common markers for hepatocytes and pancreatic cells, respectively. First is IFIC analysis showing nuclear expression of HNF4-alpha in HepG2 cells, but not in HeLa cells. HNF4 alpha is a transcription factor found in liver that plays a role in lipid metabolism and gluconeogenesis. HNF4 alpha antibody has been labeled green. Actin filaments have been labeled red with Alexaflor 555 phalloidin. HepG2 is a human liver cell line and shows a positive signal. HeLa is an epithelial cell line and serves as a negative control. In the feature below, IF analysis shows expression of insulin in rat pancreas. The insulin antibody has been labeled green. DNA has been labeled blue with DRAC5. Expression is within beta cells of the islets of Langerhans, which are the insulin-secreting cells. Epigenetic regulators and marks. As stated earlier, stem cell pluripotency is regulated and maintained by a number of cell signaling pathways leading to expression of the key transcription factors Oct-4, SOX2, and NANOG. Although expression of embryonic-specific genes is regulated by these transcription factors, a second layer of control stems from epigenetic mechanisms, which are reversible modifications made to the DNA that activate or silence gene expression. Examples of these epigenetic mechanisms that lead to chromatin remodeling are post-translational modifications to histones, such as acetylation, methylation, phosphorylation, as well as methylation of CpG nucleotides. Epigenetic mechanisms resulting in gene activation are called activating marks, and those that result in gene silencing are considered repressive marks. These marks affect how tightly or loosely DNA is wound around the histone proteins and help to regulate transcription by allowing or denying access of transcription factors to the DNA. These marks are important because although each cell within the embryo contains a complete set of DNA, only a small portion of that DNA is actively being used to maintain pluripotency. Lineage-specific genes must be turned off until needed during differentiation. Activating marks create a more open conformation to the chromatin and allow transcriptional activation to occur by providing transcription factors access to the DNA. Examples of activating marks include acetylhistone H3, lysine 9, lysine 9 and 14, or lysine 23, acetylhistone H4, lysine 8 or lysine 12, and di- or trimethylhistone H3, lysine 4. In this figure is IF analysis showing expression of the activating mark trimethylhistone H3, lysine 4 in mouse embryo. Trimethylhistone H3, lysine 4 antibody has been labeled green. Actin filaments have been labeled red with dy554 phalloidin. There are high levels of trimethylhistone H3, lysine 4 expression in this mouse embryo because transcriptional activation of embryo-specific genes is occurring. Repressive marks cause chromatin to assume a more closed conformation and do not allow transcriptional activation to occur. An example of a repressive mark is di- or trimethylhistone H3, lysine 27. In the next figure, IF analysis shows expression of the repressive mark trimethylhistone H3, lysine 27 in tissue surrounding the cartilage primordium of ribs 2 and 3 in an embryonic day 14.5 mouse embryo. Trimethylhistone H3 lysine 27 antibody has been labeled green. Actin filaments have been labeled red with dy554 phalloidin. Repressive marks will be found in the mouse embryo on lineage-specific genes that are currently inactive. A special case in the use of activating and repressive marks is called a bivalent modification or bivalent domain. Bivalent modifications occur on poised state genes and are unique to certain lineage-specific genes in embryonic stem cells. The poised state gene simultaneously contains one activating mark, usually trimethylhistone H3 lysine 4, and one repressive mark, usually trimethylhistone H3 lysine 27. As differentiation occurs along a specific lineage pathway, the repressive mark for genes in that lineage is removed, and the activating mark remains. However, repressive marks for genes in other lineages remain present. Histone-modifying enzymes. Histone modifications are carried out by a number of histone-modifying enzymes, also referred to as epigenetic regulators. For example, histone acetylation takes place through the action of acetyltransferases, HATs, such as PCAF, GCN5, and CBP. This process is reversed by histone deacetylases, HDACs, and a family of deacetylases referred to as sirtuins. Acetylation takes place on lysine residues and usually results in transcriptional activation, whereas deacetylation results in repression. In the figure on the left, IFIC analysis shows expression of the deacetylase HDAC2 in COS cells. The HDAC2 antibody has been labeled green. Actin filaments have been labeled red with alexafluor 555 phalloidin. Note the presence of green signal in the nucleus. In the figure on the right, IFIC analysis shows expression of the deacetylase SIRT1 in COS cells. The SIRT1 antibody has been labeled green. Actin filaments have been labeled red with alexafluor 555 phalloidin. Note the presence of green signal in the nucleus. Histone methylation is a major determinant for the formation of active and inactive regions of the genome and is crucial for the proper programming of the genome during development. Methylation takes place on both lysine and arginine residues and has been implicated in both transcriptional activation and silencing. This modification is carried out by the lysine methyltransferases such as SET7 and 9, SUS12, and SUV39H1, as well as the arginine methyltransferases such as MEP50 and PRMT1, 4, and 5. Reversal of these modifications takes place through the action of demethylases such as Jumanji D1B, Jumanji D2A and B, and Jumanji D3. In the figure on the left, IFIC analysis shows expression of the methyltransferase SUS12 in mouse embryonic stem cells growing on mouse embryonic fibroblast feeder cells. ESCs need to be grown on either feeder cells, as shown here, or grown in conditioned media that feeder cells have been grown in order to successfully grow in culture. The feeder cells or conditioned media provides essential growth factors that ESCs need to survive. Also shown is SUS12 expression in the neural tube of an embryonic day 10.5 mouse embryo. The SUS12 antibody has been labeled green. Actin filaments have been labeled red with DY554 phalloidin. Note the presence of green nuclear signal. In the figure on the right, IFIC analysis shows nuclear expression of Jumanji D2A methyltransferase in NCCIT embryonic carcinoma cells. Jumanji D2A antibody has been labeled green. Actin filaments have been labeled red with DY554 phalloidin. Thank you for viewing this cell signaling technology in-house training tutorial. For more information or specific questions regarding this topic, please contact the CST Marketing Department at cst_marketing@cellsignal.com.